But anyway, I'm Paul Danis with the consortium, and we're happy to have you here. This is for our Proteoform Thursday, more or less monthly seminar series, and we're excited today to have Jeffrey Huang, newly minted faculty member at Texas A&M University, and we'll introduce him in just a moment. There's a couple introductions I'd like to give. One is to thank our sponsors. We're very grateful to the support they provide us, enabled us to do these kinds of programs and build the community and do a number of projects to try to advance the technologies. So our major sponsors, Thermo Fisher, Bruker, Ciox, and Lilly, as well as our supporters, Agilent, New Objective, Pfizer, Impro, and Nautilus Biotechnologies. And also just a reminder of some of the resources we have available for people that would like to learn a little more or see some past things. We have most of our seminars as well as our workshops available online under our webpage, under resources and tutorials, or if you're a YouTube fan, you can just go to @Proteoforms and you can view a number of these there at your pleasure. They're pretty interesting, and I think some of these are really fantastic, like the vendor showcase, where some of our supporters showed some of the latest technologies they have available, as well as our recent workshop on biopharmaceutical characterization, which was really exciting. The other exciting news is some of the upcoming seminars. We have a bunch of young folks who are have really exciting work, early career people. We have Emily Bird from Leeds in April. Then we have Boris Krikl from Center of Structural Systems Biology in Germany, Mohammed Zanaidi from APAF from Macquarie, Australia, and Ana Marinho from the Oswald Cruz Foundation in Brazil. So we got most of the world covered. We got UK, Germany, Australia, and Brazil, and today we have US. So we're covering the globe pretty well there. So with that, I'd like to hand it to Neil, who's going to introduce Jeffrey.
Okay, great. Good good morning, good afternoon, um, good evening, wherever you are. And I'm excited to turn it over to Dr. Chifan Jeffrey Huang, who's been a postdoc and a student here, very much a product of Northwestern University. Uh, did his undergrad in Taiwan and then came to Milan Merksic's lab, did some SAMD work on phosphatases and using that SAMD TOF platform, and then got into Catenin biology through a postdoc with myself and Kara Guttardi, who's a Catenin expert, both in the cancer context and in the structural elements of the cell, cytoskeletal functions of Catenin and receptors that Jeffrey will get into, signaling downstream of those receptors. Without further ado, let me just reemphasize that we actually have three new professors coming out of the lab, and Jeffrey's among those at Texas A&M in College Station, together with Super Dave, who many of you know, Dave Russell for many years, and Art Lanagowski and many other great investigators there at down in Texas. So without further ado, you'll see why he competed so well nationally for that position. And Jeffrey, please just take the mic, and we look forward to hearing your story around individual INMS combined with proteoform biology.
Awesome. Let me just the video. Okay. So thank you, Neil, again for your very, very generous introduction. And I'm still currently being a postdoc with Neil and wrapping up my Catenin works here. And as mentioned, I'm also joining the Department of Chemistry at Texas A&M in the coming months. Today, I'm going to share most of my postdoc work, but I'm carrying this project with me. So this really lay out the foundation of my future work. And the title of the talk is the Catenin Phospho Code for Adherens Junction Organization. So really my passion for studying top-down proteomics and the biology of proteoform was really inspired by my training as a chemist. So as a chemist, we know that the most important things about molecular design is the structure and function relationship, and that has been the key of my concept and building my own program. So one example I remember very vividly from undergrad are the structures of these platins. So they have all the same atoms and ligands, but if you only shift them slightly from cis to trans configuration, one would be a strong DNA binding chemotherapy drug, and the other one is not effective at all. So as a protein chemist, we ask, what about proteins? Proteins are known to have a lot of modification. So under the same protein names, you would have all kinds of different modification, creating essentially different molecules and different structure. So as a chemist, we would naturally think that, oh, they must have different function. So that's where the idea of proteoforms comes from. So with this group, everyone is well acquainted with this term. Proteoform describe all protein products from a single gene. So a gene has to be transcribed and translated to protein. During these processes, there can be a lot of structural variation, such as different isoforms at RNA level and different PTMs, phosphorylation, glycosylation at protein level, giving you a spectrum of distinct primary structures of a given protein. So each distinct primary structure is essentially a proteoform. So studying protein at large scale, the best tool is definitely mass spectrometry-based proteomics. Traditional proteomics are now called bottom-up proteomics, highly rely on using trypsin or other enzymatic digestion to chop proteins into smaller peptides and identify them at peptide level. However, this would create an inference issue in terms of proteoform identification. So in this hypothetical protein and their proteoforms, we have three possible modifications that localize on different peptides. So once you chop them up, it's impossible to put the puzzle back and figure out the proteoform composition in the original sample. Therefore, there's a trend into moving into using top-down proteomics, the analysis of intact protein and fragmentation within the mass spectrometer to study proteoform level information. So in this example, again, the three proteoform without trypsin digestion, they can be directly separated by chromatography, identify their intact masses, and further fragment them within the mass spectrometer and sequence them. Therefore, you can get the accurate proteoform level assignment and even quantify them. So in summary, bottom-up proteomics give you protein level information and top-down proteomics give you proteoform level information. And top-down proteomics, that does have its own limitation. So far, we've only been able to routinely analyze species that are smaller than 30 kilodaltons. So now we know what proteoforms are, but why is it important to study biology at proteoform level? That is because proteoform describe the precise primary structure of a given protein, and therefore it is more tightly connected to cellular phenotype and therefore diseases. So in this example, we were trying to cluster and distinguish different blood cell types from blood. So these are all the cell type here on the left here, and we cluster them by their proteoforms and their proteins. And we saw one order of magnitude increase in clustering distance by using proteoforms, suggesting that proteoforms better separate and associate with the underneath biology. So now come to the question that I'm really interested in studying, which is cell-cell adhesion. Here on the left here, you're looking at an image of a lung, which is stamped with beta-catenin in green. So you can clearly see that there's adhesive junctions that provide important barrier function that separate the human body from the outside world. The junction appears to be static on the left here, but can be also really dynamic. In terms of different cellular processes, such as cell division, epithelial sheet wound repairs, the adhesive junctions has actually need to organize itself in a conservative fashion, in a controlled fashion, so that you can keep the integrity of the barrier function, but also allow the cells to move around. So there's a lot of interest in not only studying the structure of the adhesive junction, but also understanding how they're regulated in a dynamic way. So one type of the adhesive junction is the adherens junction. So in epithelial cells, you can find them between cells, which you have e-catenins that brings the cells together, that binds them, and it is a transmembrane protein, and inside the cells, it binds to beta-catenin, alpha-catenin, and then ultimately to the actin filaments. So knockout studies show that all these components, these four components are required to form an adherens junction, but how they're regulated in a dynamic way is less well understood. So here we hypothesize that junction organization is regulated by phosphorylation related to actomyosin contractility. So when the F-actin is relaxed here, alpha and beta catenin are sort of in a loose structure and not very heavily modified. But once F-actin contracts, it will rigidify the structure of alpha and beta catenin and then allow the kinase to access these phospho sites and become phosphorylated. And this hyperphosphorylated junctional complex would then promote the organization of adherens junction. So since there are so many phosphosites within alpha and beta catenin, we're interested in studying the proteoforms involved in the junction regulation and speculate that there may be a phospho-code that govern the junction architecture. So alpha and beta catenin are sitting at 85 to 100 kilodalton, which is substantially larger than what top-down proteomics routinely analyze. And one of the biggest challenge is within the separation. Larger species hard to separate efficiently with LC, and therefore you are creating a crowding in the limited N over D space as the protein charge states, modification, isotopic patterns all overlap together, giving you a spectrum looking like this, where it's just a hump of nothing and you cannot deconvolute and get any useful information out of it. On top of that, large ion also suffers from decaying. So therefore, in the Kelleher research group, in the past 5-6 years, we've been using an individual ion mass spectrometry, I2MS approach to reduce top-down spectral complexity. So this is now commercialized by Thermo Fisher under direct mass technology, or DMT. So to perform I2MS, first you have these intact proteoforms. You can limit the amount of ion injected into the orbitrap. So at a very low ion density, your MOVD spectrum will look like this, where each peak corresponds to a single ion. For each single ion, you can then monitor the signal accumulation over time, and we found that the slope of this plot would be proportional to the charge state. Therefore, you don't have to go back to the MOVD spectrum to deconvolute and get the charge state of your ion. You can determine the charge by the story plot and directly put them into mass domain, giving you a true mass spectrum. So with this approach, we demonstrate that we can profile the proteoform landscape of a recombinant GST beta-catenin protein as sitting at above 110 kilodalton and reveal multiple phosphoform. So now we want to apply this workflow to actually study endogenous alpha and beta catenin related to cell-cell adherens junction. So to do that, we have an HEK cell line that expresses an E-catenin cytoplastic fragment, which is this tail. And then it is also fused with a mirror sequence for membrane localization and a 6-MIC tag for purification. So with the tag, you can then use an anti-MIC nanobody to pull out this ECAT cyto construct together with the beta and alpha catenin that binds to it. So here we have beta catenin in red and alpha catenin in green, and on silver stand, you can also see that they are the major species being purified from these complexes. And then this LUA can then directly be profiled with individual ion mass spectrometry with all three components being resolved. So here we have ECAT cyto at 30 kilodalton, beta catenin at 85, and alpha catenin at 100 kilodalton. So zooming into the proteoform landscape of beta-catenin, first we found that we have partially resolved isotopic pattern, which is great because it allows us to do intact mass matching and calculate the observed mass and compare them to the expected mass, which allow us to confirm the first group of peaks here correspond to beta-catenin with N-terminal methionine removed and acetylation. Second group of peak, monophospho. Third group of peak, diphospho. And fourth group of peak would be triphospho. Similarly, for alpha-catenin, we were able to confirm that the first group of peaks here correspond to monophosphorylated alpha-catenin. And this is biologically interesting because we did not see any non-phospho variant of alpha-catenin, which could suggest that there may be a constitutively modified phosphorylation site that was previously unknown but important to the formation and required to the formation of adherence junction. So next, we want to correlate the proteoform landscape of alpha and beta-catenin to actomyosin contractility. So we use drugs that modulate actomyosin tension. So in the middle here is a confocal image of the cells being treated by DMSO as control. So here you can see actin in yellow aggregating between the cell at the junction and then forms a belt on top, which is consistent with apical junction formation. On the left here, the cells are treated with blebbistatin, which is the drug that inhibits actomyosin contractility. So you can see the phenotype is really clear here. The cells become larger and also flattened. So they're very relaxed without any force pulling them. On the other hand, if you treat the cells with a drug, catecholamine A, which is an actomyosin activator, you can see the phenotype is also really clear, where a lot of the cells are rounding up, a lot of junctions are broken, and you see actin being pulled toward the nuclei, which is consistent with elevated actomyosin force. Next, we can use the ecacytal construct to purify alpha and beta-catenin complexes from all these conditions. So in the middle of here is the proteoform landscape of beta-catenin that you saw earlier with up to 3 phosphorylation as a DMSO in DMSO control. On the left here, if you treat the cells with blebbistatin, we saw the reduction in the numbers of phosphorylation site and the abundances of phosphorylation. Remarkably, we saw a hyperphosphorylated proteoform landscape of beta-catenin with catecholamine A treatment and up to 10 phosphorylation within a single beta-catenin proteoform. So similarly, for alpha-catenin, we saw a hypophosphorylated proteoform landscape with blebbistatin treatment and a hyperphosphorylated proteoform landscape with catecholamine treatment and up to 7 phosphorylation. So this is consistent with our hypothesis where actomyosin contractility increase, phosphorylation increase. So with the proteoform landscape of alpha-catenin, we also make two notable observations. So the first one following the pink arrow here, where you see in all three conditions, including the blebbistatin treatment, we still see no unphosphorylated alpha-catenin. This would strongly imply that there is a constitutively modified phosphorylation site that is required for the formation of adherence junction. The second observation is to follow the blue arrows, where we saw a couple of mass shifts that does not correspond to phosphorylation, but for the sake of this talk, we'll focus on phosphorylation only. So since these phosphorylation is really, really abundant, we were able to map out all 10 phospho sites within the beta-catenin using bottom-up proteomics. Two of them fall into the ecaterin binding domain. So we have site-specific antibodies targeting these two sites. So we were able to compare their abundances across the 3 different treatments and found that their abundances increase as the actomyosin force increase. Therefore, we determined that these two are the mechano-sensitive sites. On the other hand, the N-terminal sites are traditionally associated with GSK3 beta degradation pathway, meaning that once they get phosphorylated, this molecule gets sent to proteasome for degradation. So these are traditionally not considered actomyosin or mechanophore sensitive. So we found that they exclusively show up only in the catecholinate treated conditions, which you saw earlier has a lot of broken junctions. So there's a lot of beta-catenin to be degraded. So these are definitely non-mechanosensitive sites. So with that, we propose a mechanoregulation model for beta-catenin, where the S675 and S552 are the two major mechanosensing sites, and they get phosphorylated upon high tension, whereas the N-terminal sites are associated with broken junction and proteosomal degradation. Similarly, for α-catenin, we were able to map out all 7 phosphosites. They fall into 2 domains. The first 5 sites fall into this well-established p-linker domain, which is highly conserved across different isoforms of α-catenin and different species. We also report 2 new phosphorylation sites in the actin binding domain where their function has not been described before. For the phosphosite within the p-linker domain, we do have antibodies targeting them. So again, we compare their abundances across the treatments. To our surprise, we found that the first site, the S641 sites, its abundance remained consistent across the three different treatments. So taken together with what we saw earlier in the proteoform landscape of α-catenin without non-phosphorylation variant, we determined that the S641 site is our constitutively modified site that is required for the formation of adherence junction. So similarly, we identified the rest of the sites, their abundances increase as the actomyosin force increase, so they're mechanosensing sites. So here is our mechanoregulation model for alpha-catenin, where the S641 is constitutively phosphorylated and non-mechanosensitive, whereas the rest of the sites in the p-linker domain are mechanosensing sites and get phosphorylated upon high tension. So for the newly identified actin binding domain sites, we do not have antibodies targeting them, so we don't know whether they're mechanosensitive or not. And even at a higher level, we ask if there's any crosstalk or hierarchy between the p-linker domain sites and the actin binding domain sites. So this really requires proteoform level information, but so far we've been studying this system with intact mass profiling, antibodies, site-specific antibodies, and bottom-up proteomics, none of which give you proteoform level information. So in order to get that, you really have to do true top-down and start fragmenting these giant molecules within the mass spec. It turns out that I2MS, individual ion mass spectrometry, can not only be applied for intact mass studies, but also improve fragment detection of big proteoforms, which we term this technology I2MS2. So I2MS2 was first demonstrated on MEC1 proteoforms, which are sitting at 43 kilodaltons. So similar to a regular MS2 experiment, you can use quadrupole to isolate proteoforms of interest to fragment. So you can use ETD, you can use HCD. And then the only thing that you do differently is that you use individual ion mass spectrometry, you use Story Plus to determine the charge states of each fragment to help you identify low abundant fragments within the system. So by using this approach, it was shown that you can drastically improve the sequence coverage by using from what you use an ensemble measurement, you get 3.5% sequence coverage of MEC1 proteoforms to drastically improve it beyond 50% sequence coverage. And of course, MEC1 is sitting at 43 kilodalton, is still half of the size of alpha and beta catenin. So we want to first benchmark this technology for the fragment detection for alpha and beta catenin. For beta-catenin sitting at 85 kilodaltons, we were able to achieve 25% sequence coverage with HCD fragmentation. Even more excitingly, for alpha-catenin sitting at 100 kilodalton, combined with ETD fragmentation, we were able to achieve close to 30% sequence coverage and localized the constitutively modified S641 phosphorylation sites. And of course, this is still not enough for us to look at the downstream very complicated phosphorylation pattern with up to 7 phospho sites and resolve their proteoform. So this is still ongoing work. And we're trying to improve the fragmentation and the I2MS detection. And our goal is really to go beyond 70% sequence coverage. And by the time we will able to resolve all these proteoform and study biology at proteoform level. So my passion, of course, for studying the cell-cell adhesion proteoform biology doesn't really stop at just advancing top-down proteomics and mass spectrometry technology, but I also actually want to understand what are the consequences of these phospho-code. So some of our early work within the Gattari group here is to use all these phospho-mimic and phospho-null constructs to study their behavior in cell-cell adhesion. So here we have this 4E construct where we replace 4 of the phospho sites within the P-linker domain with E mutant as a phospho-mimic and replace them as A mutants as a phospho-null control. And then we observe their behavior under confocal. This is the side view from the confocal. We found that the 4E mutant following the pink trace here localized significantly taller compared to the other mutant, which implied that it promotes the formation of adherence junction because it's taller. So one way to rationalize how did this happen is to look at how the phosphorylation within a small P-linker domain could affect the structure of α-catenin. So to that end, I was collaborating with Michael Fitzgerald at Duke University, who specialized in using an assay called stability of protein from rate of oxidation, SPROX assay, to study protein folding stability. So together, we come up with a top-down version of this assay called TD-SPROX. So to perform TD-SPROX, you have these intact proteoforms, and you subject them with a gradient of denaturant, usually it's urea, and together with hydrogen peroxide. So at any point when your proteoform unfolds, it would expose the methionine residue to oxidation. So therefore, you can create oxidation reaction curve as a function of the denaturant, and then use the transition midpoint as a simplified description of folding stability of a given proteoform. So since we're doing this in a top-down fashion, we were able to identify modifications that significantly shift the overall folding stability that are relevant. So to use this system for alpha-catenin, we took a slightly different approach because we've already have these constructs, so they're proteoformly well defined. So instead of using TD-SPROX, we use a traditional SPROX assay to get the similar information. So we hypothesized that the alpha-catenin phosphorylation within the P-linker domain would adapt a open form structure universally for the alpha-catenin proteoform. So to test that, we subject them to SPROX, treat them with a gradient of urea, and would expect to see within the phospho-mimic, it should have a shift towards a lower SPROX transition midpoint because the methionine could be more openly exposed and solvent access. So indeed, that's what we saw. Within 3 locations of methionine, we found shifts towards a lower SPROX transition midpoint, suggesting that the 4E mutant indeed adopt a more open form structure. What's even more remarkable is that all these 3 methionines are localized in important binding domains that recruit binding partners that promote cell-cell adhesion that includes beta-catenin, vinculin, and alpha-din. So this study overall tells us that the phosphorylation within the phospho domain really open up the structure and allows you to bring alpha-catenin to bring in binding partners that promote cell-cell adhesion. So this would rationalize why the phosphomimic or the phosphoproteoform can govern the junction architecture of adherence junction. So of course, this kind of study traditionally rely on individual sites that were discovered by bottom-up proteomics or site-specific antibody. So all these fossil mimic constructs use an all-on or all-off approach to turn them all on and turn them all off as an aggregation. But if you think about the proteoform landscape of alpha-catenin, there's no non-phospho variant and there's no 4-phosphoryl variants under physiological condition. So these phospho constructs, phospho mimic construct, may not be as biologically relevant. So moving forward, in my group, we will really adapt and proteoform-centric approach, where you first use top-down proteomics to identify functionally relevant, physiologically relevant proteoform for better validation of the function of these proteoforms toward better understanding proteoform-specific phospho-code. So as mentioned earlier, I'm very excited to start my own research group at Texas A&M Chemistry in the summer. So some of my projects involve in using synthetic biology approach to better help us identify large and sequence large proteoforms. So I would define my lab as a functional proteoform biology lab. So we not only want to define precise complex and protein structure, but we also want to assign their function. So in the functional end, we also want to do native mass spectrometry to go beyond just one single proteoform and look at how proteoforms interact with each other. And Neil also mentioned that from my PhD, I have a lot of interest in study phosphatases. So that carries to my independent group work, where we're gonna target some phosphatases. We know that they're regulating the cell-cell adhesion systems, but they're largely neglected because most people focus on kinases and how they add phosphorylation. So we're gonna look at the dark side of phospho control. With a niche area that is very understudied. So the key technology in my lab would be top-down proteomics and native mass spectrometry. The biological questions that we're studying are cell-cell adhesion and phosphatase biology at proteoform level. We will involve synthetic biology approach and structural biology approaches. So I'm currently accepting students from Texas A&M's chemistry graduate program, and there will also be one postdoc opportunity coming up earlier next year. So if you know anyone that are interested in doing a PhD or postdoc, please do not hesitate to reach out. And with that, I also want to acknowledge, of course, my advisor, Neil Kelleher, who really brought me into this top-down proteomics field and really established this exciting program. And Kara has been my collaborator for the past 8 years in the Catenin biology. We not only study the cell-cell adhesion, but we also have some cancer signaling stories that if you want to check out, please go check out the paper. Michael Fitzgerald on the structural biology side with the Sprox assay. Tao Su is really a very great grad student in the Kelleher Research Group, now graduated, but really is the driver of the I2MS2 study. Jin Quinn and NF from the Caricaturity Group. My mentor, Rafael Mulani, who really taught me hand in hand everything about top-down proteomics, and Yuzo from the Duke University on the Sprox collaboration. Everyone in the Kelleher Research Group, here are my funding sources. And again, special thanks to the consortium for top-down proteomics for the invitation to share some of my work. With that, I'll be happy to take questions.
Great. Thank you, Jeffrey. That's fascinating. Great work. We do have a question. Mike Senko has a question. It said in slide 29,
Yes.
there seemed to be some structure in each of the phosphoforms. Do you have any thoughts on what this might be?
Yes. Do you guys still see the sharing?
No, not right now.
Okay.
Yeah.
You said 29?
Yes, that was what he said.
Yes. So we know that the monophosphorylated is definitely the constitutively modified sites is 641. These two, we have a very solid guess. So our hypothesis right now is that the phosphorylation of alpha-catenin is sequential. So since this is the priming sites, we hypothesize that it might be kind of a ratchet kind of feeling where as the phosphorylation open up the structure, it kind of allow kinase assays to the access to the downstream sites. So our hypothesis currently now is that we know that monophosphorylated would be this side, and then it's kind of start progress a lot in terms of proximity. So we currently don't have solid data supporting this, but this is our current working model, and that's why we're still pushing forward to do the MS2 level identification to really resolve this.
Yeah,
Okay,
no,
thanks.
Jeffrey, it's Mike. I was talking more. There seems to be something underneath the second and third phosphorylations,
Mm-hmm.
Yeah,
and
Oh, you mean because it's broader, right?
right.
yeah,
And Jeff
Yes.
so
Silva picked up on that same thing. Yeah.
yeah,
Yes.
that first phosphorylation is like incredibly clean,
Yes.
and then the second and the third, there seems to be something else underneath it.
I agree. So if you look at the blebbistatin treatment, I think there are some modification that could be involved, other modification that could be involved. So blebbistatin kind of separate that, but I think under physiological condition, they kind of just all or are modified together. So there can be something else that are involved that we're not able to resolve
And
at
it
MS1 space.
isn't it more consistent with an additional mass beyond the 80? Wasn't it in the review of the paper we ran around this where it could it have been oxidation in addition?
Yes, we we did match some oxidized form. So here on the right here could be some modification, but on the left here is the solid uh, plus 80 peak here.
So
So
it's
one
complete.
possibility could
Oh,
be
good.
oxidation, but can also be phosphate potentially.
Yeah, but it's so reproducible. You know, phosphate is this artifactual addition of 98, not 80.
Mhm.
And so,
Yes.
but the first one's really clean. You usually see the phosphate adducting to everything. Um,
Yeah.
so that's why I was thinking, and now this is totally conjecture, but there is like the HIF1 alpha is an oxygen sensor. There is a hydroxylation or an oxidation event in response to reactive oxygen. But that,
Mhm.
but great eyes, everybody picking up on that. It's reproducible, and so it confuses us to this day.
Well,
Yeah.
let's go to, I think Jenny's had her hand raised. The arm's getting tired up there in the corner of the
Yeah.
screen, I can see. So why don't you go ahead, Jenny?
Okay, great. Thank you. And congratulations, Jeff, on your position at Texas A&M.
Thank
We'll-
you.
We will be neighbors and rivals, but that's fine. And my question was, the MSMS for the direct mass technology is super exciting, and I'm wondering, do you see any isotope patterns? Because often we seem like we're using those to fit our fragment ions and confirm them. So how do you deal with that at the MSMS level?
Yes, we do see isotopic patterns. So most of the under 50 kilodalton species, even in the MS2 space, can be baseline resolved with isotopic pattern. But because it's decharged, so they are just like 1 dalton apart. But we do see isotopic pattern, and that allow us. So all of these yeah, I don't have a zoom in right now, but all of these fragments that you see with these diamonds, there are multiple diamonds on the same peak, which if you zoom in, that would be the resolved isotopic pattern.
Thank you.
Great there. Jeff, do you have a question there?
Yes, great talk. Thank you so much. I do have a question about just to hear your thoughts about the utility of antibodies, commercial antibodies,
Mhm.
um, that are targeted to specific PTM sites in terms of their utility for these types of experiments to be able to fractionate or enrich for subsets of the proteoforms to do further characterization. Um, just curious to hear what your thoughts are on that and whether or not you think these down the road could be useful tools to sort of tease apart the contributions from each of the different proteoforms.
I completely agree. One of the biggest challenge that we have is really to purify the proteoform and really separate them because at this mass range, we're doing direct injection, but even if we do LC, usually all the alpha-catenin proteoforms, they elute altogether. So there's no way to cleanly separate them. And especially they're charged at 100 plus mass. So when you do quadruple isolation, they're often you can just not you just cannot separate them. So yes, antibodies are really important. I've not done a fossil-specific antibodies IP, so I don't really know if we can really separate them that way, but that adds another layer to how to separate them. So fossil antibodies would definitely be modification specific antibody could definitely be utilized there. And there are a lot of known modifications to alpha-catenin that we did not pick up here. Also, potentially maybe their abundance is just way lower than 1% we're not seeing. So down the road, we are also interested in looking at some rare modifications that are known to the literature that has specific functions and potentially pull them out, enrich them using antibodies.
I'm going to provide my email. If you could reach out, I'd like to sort of talk to you more about this because we may be able to have some reagents available for these types of applications.
Yes, and also the other point is using yeast, right? I think that is also an approach that I'm taking. I kind of make it a little vague on what synthetic biology approach that we will be taking. So there are a lot of construction of these different proteoform specific constructs. I think within a cleaner system would be much easier to work with. So yeast, E. coli, and C. elegans potentially are all approaches that I'm considering.
Very good.
Okay, thanks. Eric, did you have a question there?
Yeah, I do have a question. I was wondering how long is ion accumulation time before performing MSMS?
Mm-hmm. So far, we've been using so these are really giant molecules. So the sequence coverage that you saw here were performed on Orbitrap Eclipse. I2MS2, we collect them for 1 hour. I think there are a lot of exciting things that's happening within the instrumentation space. We've tried on Orbitrap Ascend, where like you have improved ion survival time, you start seeing the bigger ions, and also they survive better. That could shorten the reaction and collection time. So in terms of instrumentation, it's gonna get improved. But this was collected probably over a year ago. Was performed on Eclipse, so we use 1 hour collection.
I see. Thank you.
Great. I had a question on you looking forward. You mentioned a few areas you're going to focus on, but I guess two-part question. One is I didn't see sort of technology development within part of that. Is that implicit in part of your program? And then if so, like what are kind of or you just relying on Mike Senko to make cool new tools, you know?
Well,
And if that is the case, then what are kind of the main thrusts of technology needs that you have at this point?
yeah, I kind of it was kind of a strategy when I was on the job market. I was kind of proposing things that I know I can do it in-house on my own uh, without involving others because I don't know what kind of assets will I get have
Mhm.
the luxury to have. So yeah, moving forward, I'm definitely excited in instrumentation, and specifically at Texas A&M, there's already standing collaboration with Thermal on using eye mobility and also combining them with DMT. So the position at Texas A&M really allowed me to like kind of broaden that instrumentation aspect. There would be some exciting things that I would be doing with Dave and aren't already uh, in movement. Yes.
So what would you say like the one or two hottest areas you need technology to evolve to really get to your bottom line? What are is it sensitivity? Is it separations, antibodies? What are kind of the couple technology advances that you would love to see happen in the next 5 years?
Honestly, bioinformatics, I feel like that is really, really, really the key because when you are thinking about things this big, 100 kilodalton, so that means you have over 800 cleavage site for amino acids. And if you have three kinds of cleave form involving UVPD, ETD, and HCD, you have all these crazy kind of overlapping fragments that you need to resolve and somehow tease out how true they are. So I think bioinformatics is really going to be the biggest challenge to interpret these very complicated data.
Oh, great, great. Any more questions or comments for Jeffrey? Great. If not, then thank you again, Jeffrey, for sharing your work. Pretty exciting stuff, and we all wish you best of luck at A&M. So
Thank
thank you
you.
again, and I wish everyone a good day. Okay, bye-bye now.